Hi friends, in this we are going to discuss about the list of various biometrical techniques used in the crop improvement. First we need to know what's biometrical techniques. The various statistical procedures which are employed in biometrical genetics are called as biometrical techniques. And in plant breeding research, various biometrical techniques are used for improvement of yield and its contributing traits. On the basis of their applications in plant breeding, biometrical techniques can be divided into four different groups. That is, assessment of variation or variability, a selection of allied genotypes, choice of suitable parents, and breeding procedures, and fourth one, assessment of stability of genotypes. First, coming to the first one, assessment of variability. In this, success of genetic improvement in any characters depends on the nature of variability present in the gene pool of the characters. Hence, the assessment of existing variability for any character present in the gene pool of a crop species is at most importance to the breeder for starting a judicious plant breeding program. And biometrical techniques for systemic assessment of variability are uh, like a measure of dispersion and components of genetic variance and metroglyph analysis, d-square statistics, and principal component analysis. In this, first the technique which is used and the genetic information that we are going to get after the using these techniques, like by using the measures of dispersion like range, standard deviation, variance, standard error, coefficient of variance, we are going to get phenotypic variability. We are going to get what is the phenotypic variability means. Range means what is, for example, range means what is the range of that traits, like small to like 1000 to 200, 1 to 10, like range. We are going to set the range. And standard deviation, and what is deviation from, if suppose 10 is a 10 is optimum one, that what is the deviation from, standard deviation from, like below and above, standard deviation. And variance, standard error, what is standard error, and coefficient of variance. For this all these if we calculated these things, we are going to get the phenotypic variability between those, between the genotypes or anything what under the consideration of studies. And component of genetic variances, uh, phenotypic, genotypic, environmental variances, and phenotypic covariance, genotypic covariance, environmental covariance. By finding these things, we can get heritability and genetic advance and gene action involved. Like after finding using these techniques, we are going to see what is heritability, what is the percentage of heritability, what extent the trait is heritable, that is means one generation to next generation, what is heritability we can see. And genetic advance, is there any helpful for next further generation, genetic advance helpful we are going to see. And gene action, different types of gene action, additive gene action, dominant gene action, these are all gene action we are going to study, we are going to obtain by these by all these, by all these techniques. By using these techniques, we are going to find these all techniques. And metroglyph analysis, it is a type of analysis we'll discuss in the next videos. Please tune for this day, we'll give brief right here. And with the help of this biometrical technique, we are going to get variability and diversity, how much variability is and how much diversity is there. Variability, simple sense, means variability means comparing between two genotypes or two species genotypes. We are going to say variability, what is the difference between. Diversity means it's a large population. In this population, we are going to see how much diversity is there, how much difference is there. We need to know the difference between variability and diversity. And d-square statistics, we are going to get the genetic diversity. And also principal component analysis, also we are going to get genetic diversity. As I said, genetically diverse, how much diverse is there in a population. And selection, and coming to second, selection of allied genotypes, it's also most important one, that the selection of superior genotypes from the breeding population is at most important activity in all plant breeding programs, because we need to find the we need to select the superior genotypes, superior genotypes from the population. It should be for the breeding procedure. For this, to select the superior genotypes, we use correlation analysis, path coefficient analysis, and discriminant function analysis. By using correlation analysis, correlation means what is the correlation between correlation. Co means at a time. The relation analysis. The at a time correlation. What is the correlation between two traits? We can study by this technique. We are going to get character association. What is character associated with that correlation? And path coefficient analysis, we are going to get the information about cause and effects. What's the cause and what's the effect of that? And discriminant function analysis, selection criteria. And by the technique called discriminant function analysis, we are going to get the selection criteria. We are going to obtain selection criteria. Coming to the choice of parents and breeding procedures, it's also one of the most important that choice of the parent and breeding procedures is a must. Like first, if any breeding program has to start, we should first select suitable parents for the hybridization program. The all breeding is almost depends on the hybridization. Hybridization is one of the most important process. Selection of the parents is at most important. And after selecting the parent, we need to say which breeding program we have procedure we need to follow. This is also one of the most important. For this, analysis of several single crosses and diallele cross analysis and partial diallele analysis, line to tester analysis. By this technique, all these techniques, with the help of diallele analysis, diallele cross means the crossing of every parent in every possible crosses diallele, and it's like a parents both the lines will act as a male as well as female. All cross combinations are studied. By this diallele cross analysis, we are going to get GCA, SCA variances and effects and additive and dominance components. We are going to study all these stuffs. And partial alleles means one of the parent is kept common. One of the parents is kept common. One of the parents is kept common and other is changing. And only we can find only GCA general combining ability, because if one of the parent is kept common means that only how much the one of the parents, one of the genotype is combines with other. GCA means general combining ability, SCA specific combining ability. GCA effects and GCA variances and the additive and dominance components we can study. Line neutral tester analysis, by this we can study GCA and SCA variances and effects and additive and dominance components. Analysis of three-way and double crosses. For analysis of three-way and double cross, we are going to study triallele, quadrillele analysis, additive, dominance, and epistatic variances. Analysis of individual crosses. In this, we are going to analyze several single crosses here. First one. But in this, analysis of individual crosses. Generation mean analysis, biparental cross analysis, triple trace cross analysis. In the generation mean analysis, we are going to get the additive, dominance, epistatic variances, and biparental cross analysis, we are going to study about the additive, dominance, variances, and triple trace cross analysis, additive, dominance, variance, and presence or absence of epistasis. It's a more important. It's one of the important presence or absence of epistasis we are going to find. Assessment of variability. If one should know how much adaptability of the plant is going to take place for in a particular environment, to how much the varietal adaptability we are going to study by the with respect to concerned environment. Stability analysis model, that is phenotypic stability, we are going to get what is the phenotypic stability is there. Example for stability analysis models like parametric models and regression model, non-parametric model, and multivariate analysis And these are all stuffs we can multivariate analysis and non-parametric analysis. These are the stability analysis model. By this we can study the varietal adaptability. Thank you. Please subscribe our channels. Please, stay tuned for other different models like d-square, metroglyph analysis, component analysis, and correlation path analysis. These are all study. Please watch our other videos. If you need any, please comment and share the video. Thank you.